Welcome back. Welcome to Chapter 3, The Cell. This is Section 1, Spontaneous Generation. And then we go through the foundations of modern cell theory, unique characteristics of prokaryotic cells, and unique characteristics of eukaryotic cells. Introduction. Life takes many forms, from giant redwood trees towering hundreds of feet in the air to the tiniest known microbes, which measure only a few billionths of a meter. Humans have long pondered life's origins and debated the defining characteristics of life. But our understanding of these concepts has changed radically since the invention of the microscope. In the 17th century, observations of microscopic life led to the development of cell theory. The idea that the fundamental unit of life is the cell, that all organisms contain at least one cell, and that cells only come from other cells. Despite sharing certain characteristics, cells may vary significantly. The two main types of cells are prokaryotic cells, lacking a nucleus, and eukaryotic cells, containing a well-organized membrane-bound nucleus. Each type of cell exhibits remarkable variety in structure, function, and metabolic activity. This chapter will focus on the historical discoveries that have shaped our current understanding of microbes, including their origins and their role in human disease. We will then explore the distinguishing characteristics found in prokaryotic and eukaryotic cells. So, Section 1, Spontaneous Generation. Learning objectives. Explain the theory of spontaneous generation and why people once accepted it as an explanation for the existence of certain types of organisms. Explain how certain individuals, Van Helmont, Redi, Needham, Spallanzani, and Pasteur, tried to prove or disprove spontaneous generation. Humans have been asking for millennia, where does new life come from? Religion, philosophy, and science have all wrestled with this question. One of the oldest explanations was the theory of spontaneous generation, which can be traced back to the ancient Greeks and was widely accepted through the Middle Ages. The theory of spontaneous generation. The Greek philosopher Aristotle, 384 to 322 BCE, was one of the earliest recorded scholars to articulate the theory of spontaneous generation, the notion that life can arise from non-living matter. Aristotle proposed that life arose from non-living material if the material contained pneuma, vital heat. As evidence, he noted several instances of the appearance of animals from environments previously devoid of such animals, such as the seemingly sudden appearance of fish in a new puddle of water. The theory persisted into the 17th century, when scientists undertook additional experimentation to support or disprove it. By this time, the proponents of the theory cited how frogs simply seemed to appear along the muddy banks of the Nile River in Egypt during the annual flooding. Others observed that mice simply appeared among grain stored in barns with thatched roofs. When the roof leaked and the grain molded, mice appeared. Jan Baptista van Helmont, a 17th century Flemish scientist, proposed that mice could arise from rags and wheat kernels left in an open container for 3 weeks. In reality, such habitats provide ideal food sources and shelter for mouse populations to flourish. However, one of Van Helmont's contemporaries, Italian physician Francesco Redi, 1626 to 1697, performed an experiment in 1668 that was one of the first to refute the idea that maggots, the larvae of flies, spontaneously generate on meat left out in the open air. He predicted that preventing flies from having direct contact with the meat would also prevent the appearance of maggots. Redi left meat in each of 6 containers, so there were 2 of each of these, or 1 replicant for each condition. 2 were open to the air, 2 were covered with gauze, and 2 were tightly sealed. His hypothesis was supported when maggots developed in the uncovered jars, but no maggots appeared in either the gauze-covered or the tightly sealed jars. He concluded that maggots could only form when flies were allowed to lay eggs in the meat, and that the maggots were the offspring of flies, not the product of spontaneous generation. Clinical focus, Part 1. Barbara is a 19-year-old college student living in the dormitory. In January, she came down with a sore throat, headache, mild fever, chills, and a violent but unproductive, no mucus, cough. To treat these symptoms, Barbara began taking an over-the-counter cold medication, which did not seem to work. In fact, over the next few days, while some of Barbara's symptoms began to resolve, her cough and fever persisted, and she felt very tired and weak. What types of respiratory diseases may be responsible? In 1745, John Needham, who lived from 1713 to 1781, published a report of his own experiments in which he briefly boiled broth infused with plant or animal matter, hoping to kill all pre-existing microbes. He then sealed the flasks. After a few days, Needham observed that the broth had become cloudy and a single drop contained numerous microscopic creatures. He argued that the new microbes must have arisen spontaneously. In reality, however, he likely did not boil the broth enough to kill all pre-existing microbes. Lazzaro Spallanzani, 1729 to 1799, did not agree with Needham's conclusions, however, and performed hundreds of carefully executed experiments using heated broth. As in Needham's experiment, broth in sealed jars and unsealed jars was infused with plant and animal matter. Spallanzani's results contradicted the findings of Needham. Heated but sealed flasks remained clear, without any signs of spontaneous growth, unless the flasks were subsequently opened to the air. This suggested that microbes were introduced into these flasks from the air. In response to Spallanzani's findings, Needham argued that life originates from a life force, Aristotle's idea, that was destroyed during Spallanzani's extended boiling. Any subsequent sealing of the flasks then prevented new life force from entering and causing spontaneous generation. Disproving spontaneous generation. The debate over spontaneous generation continued well into the 19th century, with scientists serving as proponents of both sides. To settle the debate, the Paris Academy of Sciences offered a prize for resolution of the problem. Louis Pasteur, a prominent French chemist who had been studying microbial fermentation and the causes of wine spoilage, accepted the challenge. In 1858, Pasteur filtered air through a gun cotton filter and, upon microscopic examination of the cotton, found it full of microorganisms, suggesting that the exposure of broth to air was not introducing a life force to the broth, but rather airborne microorganisms. Later, Pasteur made a series of flasks with long, twisted necks, swan neck flasks, in which he boiled broth to sterilize it. His design allowed air inside the flasks to be exchanged with air from the outside, but prevented the introduction of any airborne microorganisms, which would get caught in the twists and bends of the flask's necks. So a trap, essentially. If a life force besides the airborne microorganisms were responsible for microbial growth within the sterilized flasks, it would have access to the broth, whereas the microorganisms would not. He correctly predicted that sterilized broth in his swan neck flasks would remain sterile as long as the swan necks remained intact. However, should the necks be broken, microorganisms would be introduced, contaminating the flasks and allowing microbial growth within the broth. Pasteur's set of experiments irrefutably disproved the theory of spontaneous generation and earned him the prestigious Alhumbert Prize from the Paris Academy of Sciences in 1862. In a subsequent lecture in 1864, Pasteur articulated Omne vivum ex vivo, life only comes from life. In his lecture, Pasteur recounted his famous swan neck flask experiment, stating that life is a germ and a germ is life. Never will the doctrine of spontaneous generation recover from the mortal blow of this simple experiment. To Pasteur's credit, it never has. And that brings us to the end of Section 1. Tune in next time for Section 2, Foundations of Modern Cell Theory. Until then.